Without further ado, I will introduce Dr. William Chen, who we are so excited to have with us here today. He is a brilliant scientist, professor and director of NTU's Food Science and Technology program. He has so much experience in developing food tech innovations that are focused on reducing food waste and powering a circular economy in Singapore. And as many of you know, a critical part of ensuring that alternative proteins can actually sustainably feed the world will mean that we have to be very creative and resourceful with our inputs. And that's why I'm so excited to learn from William today about how to use agricultural waste streams to power microbial fermentation. So that's about all. William, you can feel free to share your screen now, and I'll turn it over to you to get started. William's seminar will last approximately 40 minutes, and then we will have an additional up to 40 minutes for Q&A. So make sure you take advantage of the Q&A section down below to put in your questions for William. And this seminar, as always, will be recorded and put on our YouTube channel after our seminar concludes. Awesome. Thank you so much. William, feel free to take it away. Oh, sorry, we're having, I think you might be muted.
All right.
Oh, we're still having a little bit of trouble hearing you.
All right, let's try again.
Okay, that's perfect.
Is it okay now?
Yeah, we can hear you now. Sorry about that. I think that was me.
All right. Hi, good evening for friends from the North America, and good morning for friends from Asia. I think in Europe it's still quite, it's only early in the morning. So, all right. Thank you, Amy, for the very kind introduction, very kind words. It's an honor to be giving this webinar. My name is William Chen. I'm from the Nanyang Technology University in Singapore. So today I would like to use one example of our research activity to illustrate how we push towards food waste reduction and then create this circular economy. But I also would like to share with the audience about the philosophy of moving towards this direction, and beyond this, what else we have been doing. Okay, so as I'm getting old, so I tend to forget things very fast. So before I forget, I would like to acknowledge the student who has been involved in this work. Her name is Sajindra Kure, holding the award, a cert, and she was from Sri Lanka, and she had done this work during the course of her PhD. And the award that she's holding is actually delivered, hand over to her by this Professor Francis Arnold, who is actually the Nobel laureate for chemistry 2 years ago, I think. Yeah. So that is in Singapore, actually. For those friends who have not been to Asia or Singapore, I'd like to introduce a little bit of a geography. Oh no, this is not the geography. So the problem here we like to see is actually taken from the a website to say that we need to really take care of the earth, not just ourselves. This is the situation, what we call Earth Overshoot Day. Basically, what you see from the bottom to the top is that we are abusing Earth increasingly, and so much so that our own survival may be in doubt, right? So there are a lot of things that we can do, but we'll talk more about food today. So for those of you who have not been to Singapore, this is a map on the left-hand side. This is a map of the Southeast Asia. You probably will find it very hard to locate Singapore, but this is at the in between the two green color island you see here. We are at the tip of this peninsula, and very close to Malaysia. On the right hand side, what you see is actually the map of Singapore, actually about MRT network, and our university is located on the west end of Singapore. How big is Singapore? Well, we are 700 kilometers square, so I think it's smaller than any city most of the city in the US or Canada. So what is a big picture behind my talk today? Well, this is about Singapore. SG stands for Singapore. In Singapore, we like to use acronym. Everything has an acronym. SG stands for Singapore. So we talk about Singapore food security, now and future. This is a survey done by the magazine The Economist. In 2018 and even last year, we were ranked top in the world in terms of food security index. You may wonder, how can that be, right? Because Singapore is not an agriculture country, and we don't have any natural resources. Well, if we look at the criteria, right, assessment criteria, they are mainly based on three. One is affordability, second one is availability, and third one is quality and safety. And for those who have been to Singapore, you will see that we actually enjoy eating. Eating is a part of our culture, and we really very much enjoy eating. So the food is very much available, very affordable, and very safe. But the problem along here is that we're actually not sure how much we should eat. We enjoy eating so much that we don't take food security very seriously. Any concern around food security, we don't take it very seriously. But there are underlying risks. For example, we import 90% of the food from over 180 countries. And in the world, you know, there are only 190 countries around in the whole world. So we actually virtually import food from almost every country. This diversification actually lay out the buffer and safety network for our food availability. However, there's a number of risks that we need to bear in mind. First is climate change, global warming, which one way or another will affect the agriculture primary production yield, and in turn would actually affect the ability for the agricultural country to export food. The more acute example we have seen is actually the current COVID-19 pandemic, which has led to this collapse of supply chain of goods, including food. So with this as a backdrop, there's an urgent need to push for Singapore food security, right? So Singapore government has set out this vision and mission to achieve a 30% nutrition requirement from local production in about 10 years' time. The important part here is about nutrition requirement. We're not talking about volume of production, right? But that's beyond the scope of today's talk, but it's related to the big picture. What are the challenges facing Singapore food security? On the left-hand side, you see the same map of Singapore. The green color indicates the farming land. So in the early 60s, when Singapore was just gained independence, we have about 25% of farming land producing sufficient food to feed the population. The then Prime Minister Lee Kuan Yew did not think this is the way to go, because we should strive the economy since we do not have much of natural resources. So to this, there's a trade-off. So trade-off is that we will start importing food and then clear the land for industrialization and urbanization. So much so that you can see that over the years, we have a much less land for farming, Now we have about less than 1% of farming land. The second issue is the food waste. So as I mentioned, we really enjoy eating, and the food is really good here. But we generate a lot of food waste as we don't take food security for very seriously. Every year we generate, the food waste we generate can fill up about 1,500 Olympic-sized swimming pools. Same volume of plastic waste has also been generated every year. So this is a serious issue. The other one is, the third one is aging population. Singapore is a very hard-working place. People spend a lot of time in office, lesser time to date or to get married and to have children. So it's a very fast aging population, right? So I look at these three areas. They are interconnected and all part of Singapore food security, and by and large can be applied to other countries too, right? So how to enhance Singapore food security? Primary production, even with less than 1% of farmland, we can go vertical, right? So this urban farming, vertical farming is very much on the radar screen in the local space. And we already see vegetable produce on the local vertical farming in the supermarket. They taste equally good and slightly more expensive, but with demand will drive supply. So with higher demand, I think the price will come down. Yeah, and the urban farming has served also another purpose because consumers now have new lifestyle. They want fresher, nutritious food, and urban farming will actually compensate the lack of these two elements from the traditional farming, which tend to be far away from the urban area. The second one is processing technology. So here we talk about how to extend shelf life to reduce food waste. And today I will focus on how we recover nutrients from food waste, and so that we can reconnect back to the food chain and then ultimately reduce the burden of food import to Singapore. Nutrition will not be part of the topic today, but that's related. We may probably discuss this at a later stage. So at my university, we are only 29 years old, although we are ranked 11 or 12 in the world. We are only 29 years old. I'm twice as old as my university, and so we, as such, we do not have much credential in the agriculture sector. We do not have a farming school. We don't have a nutrition school, but we have a lot of engineers and scientists and communication colleagues who can contribute to this interdisciplinary area, that is enhanced processing technology. So we moved into this area 5 years ago, when we first started the food science and technology program. And now it has gained so much attention that it become a government's top priority to reduce food waste among other general waste. So this is in a nutshell what we have here. We have two elements, as you can see. On the left-hand side is more on education, or right-hand side is on research. In education, we partner with a Dutch university called Wageningen University Research. They are ranked #1 in the world in the area of food science and technology. So we are very much into talent development together with them. But in terms of research, we have sort of started this various initiatives on our own. Basically, because if you go to the Netherlands, you see that everyone is 2 feet taller than me. I'm 6 feet tall. So that is a reflection of the nutrition and diet, the difference between the Netherlands and Singapore. So we thought that we should do something by using the innovative mindset. We should do something about Singapore food problem. So we have developed in-house capabilities. You can see at the bottom 3 areas on the right-hand side, we have created this food circular economy by developing fermentation, gut health, green processing, natural food preservative, and zero waste food processing technology. And these technology are very simple and cost effective. Because at the time, my university was asking me to develop education partnership, so I did not have much funding to run the research. So we had to find ways to develop a research to give our education some identity, and using very little funding. Of course, on the hindsight, when we look back, it's actually a blessing in disguise because industry loves our simple innovations. So as a result, we have a lot of industry engagement and also government engagement. As you can see, our partners in FST, the number of industries partnering with us, including some US companies. We have also some deep engagement with Singapore government. So as I mentioned just now to Amy, we just yesterday we just managed to bring in a huge funding for our food research around US 20 million. So this is compared to $30,000 fund that I had to start our food research. So within 5 years, things can change dramatically. All right, so these are coming closer to the topic of, I was told by Amy that I should be more technical today. So I'm moving towards that part. So you can see that we generate a lot of food waste, general food waste. We also have a lot of side streams. Side stream meaning that they are from the food processing industry. For example, soybean residue that you see okara. Okara is a Japanese word, stand for noble husk. So this is a soybean residue. Soybean residue generally is a side product from bean curd manufacturing. Spent grain. Spent grain is in the beer making, also in some of local beverage called Milo. So we use barley grain actually. Once the nutrient is taken out, squeezed out from the barley grain, the rest we call it spent grain, right? And the other one is waste cooking oil. So why I highlight these three side streams? Because side stream has an advantage because they are homogeneous compared to general household food waste, which we are moving into now. These side streams valorization much higher potential in terms of scale up for industry application, right? So now brewer spent grain, as I mentioned, this is a barley, right? So we let it germinate, and then the liquid from the germination contain a lot of sugar, and they will be feed the yeast to make beer. This is simple in a nutshell. What is how the spent grain is generated from a brewery. And so you can see on the right hand side that we generate a lot of BSG. BSG stands for brewer spent grain, right? And people drink a lot of beer in Singapore also. Yeah, although it's more expensive here, but we still drink a lot. But you can see that 20 kg of BSG can be generated on this for making 100 liters of beer. So it's a lot of BSG generated. So what do we do with brewer spent grain in Singapore? As I mentioned, we have only 1% of farming land, so only 2 chicken farms. So there's no way we can channel this waste to animal farm. In a bigger country with more animal farms, these brewer spent grain as well as soybean residue can be channeled to as animal feed. But here is not possible. So what we do, we burn it, right? Simple solution, we burn it together with other food waste. But the burning is cause a number of issues. First one is energy consumption. Second one is the ashes that we send for landfill. We are reaching the full capacity for the last island, the landfill, using landfill. That is the capacity we fill up by 2035. So we also lose a lot of nutrient that remains in this side stream, right? So this is you can see moving closer to the topic. So this is what people do is spent green, right? There are some activity that try to recover this nutrient from the brewer spent green, but not significant enough, right? So what we have decided to do is to use this is a composition of a brewer spent green. You can see that it's still very much rich in protein, for example, a lipid, right? We do this analysis before we embark on our research initiative. Interestingly, there are also a lot of fiber that cannot be removed by fermentation, because basically these are the fermentation is actually hydrolysis, breaking down all these soluble components from the raw materials. So waste valorization, right? So waste valorization, there are many ways: physical, chemical, biological. So our purpose, motivation is to find ways to valorize side streams. I'm very reluctant to call them waste because they're so nutritious. How we find it nutritious? Because you know, this side stream, they get spoiled within 2 hours at room temperature. But Singapore is very hot. I think in the winter in North America, it will not get spoiled so fast. But this is an indication, the fast spoilage is, to me, is an indication of a high nutrient content. So microbes love it, right? Microbes will just attack this nutritious side stream. So instead of running away from these smelly side streams, we actually try to develop a method to valorize it. And there are a lot of spillover from our fermentation technology. For example, as you know, in animal farming, people use a lot of antibiotics to boost the production yield. These are many issues, not that, in addition to antibiotic migration to human to consumers, there are also issue of antibiotic resistant bacteria. So our fermentation is wonderful because after the fermentation, all the side streams will not get spoiled anymore. So this is actually in line with a new practice in the aquaculture and animal farming, that is using probiotic to replace antibiotic in this farming practice. So you can see that we're trying to move towards biological, environmental friendly practices. Yeah. Coming back to the technical side. So fermentation, this one is, we did not invent fermentation. So we try to, the food is so interesting because it's a space that everybody can contribute, not just in by the passion, but also by the skill set, right? So we can see that the fermentation can be applied to bioethanol production in the area of bioenergy. That was about 10 years ago. Fermentation is particularly interesting and popular in Asia because this is one way to preserve the food. And like in Korea, we have kimchi. In Japan, we have miso. In Singapore, we have called nattos. It's a local type of food that also using fermentation to sort of ferment the beans, right? It's actually quite tasty. Yeah. So SSF stands for solid state fermentation. So two types, mainly two types. The other one is submerged fermentation. So today we're talking about the solid state fermentation. There are also important aspect is the monitoring these bioprocesses, because once the lab skill technology move up to the industry pilot skill and the industry side, monitoring of performance is very important. So we also develop metabolomics, but due to time constraint, I will not move into the details of metabolomics analysis. Okay, so research background. Okay, so first we analyze the suitability and the result of fermentation applied to the BSG. Second, we see the application, because no point just reduce spoilage and do nothing about the product. So we have two areas for the application. One is to see whether it can be developed into a beverage for consumers. Second is to replace some of the culture medium used in the biology industry and urban farming. For the biology industry, today I'll focus on the biology industry application. And the remaining residue actually can also be converted into something very useful. This is something we are very proud to be working with a local energetic startup called Autopack. So they are really enthusiastic about this circular economy and the main use of these side streams to develop some useful biodegradable product. But that I will leave them to talk more in the future. Today I'll focus on the nutrition recovery aspect. Okay, so valorization of this brewer spangri. So as you know, a lot of food waste can be recycled into compost, fertilizer, but to me, it is more like a recycling. What we are doing here is to find ways to upcycle the side stream. The idea is very similar to Singapore, how Singapore has developed its economy. So we import the crude oil, we process it, convert into high value fine chemical, and then we export it out. So along the way, we boost our economy. Likewise, we will find ways to upcycle this side stream, like for example, nutrient and so on, and fine chemicals too. In this case, fine chemical refers to food additive, which is not part of today's topic, but you will see at the end, I will list out a few representative publications that you can maybe refer to after this webinar. Okay, all right. So I have talked so much, so we're still not into the technical part. Now I will move in, right? Okay, so the idea here, the aim is to use fermentation to extract nutrient, right? So what is novelty? Novelty is first we develop this upcycling process, and it has a wide range of application, as I mentioned, for urban farming, for replacement of culture media and biologic industry, and also for biodegradable product development. Okay, this is how it looks like. What you see on the left-hand side is actually in the brewer spangri in the petri dish, and we use a full-grade fungi. We apply solid-state fermentation. Although the color does not look very pleasant, but it doesn't smell bad. It really doesn't smell bad. And the solution after the solid-state fermentation, when we wash out this fermented product, the solution is actually quite fruity, right? I taste all the innovation product from my program, because otherwise I find it very hard to promote my product that I don't even know how they taste like. So in this case, it doesn't taste like beer, but it tastes very fruity. Yeah. Okay, so this is what you see. Well, this is a technical part. So before and after, you can see that the fermented product, right? The sugar level goes down, and the fat level goes down, and actually what is not shown here is actually the free amino acid level goes up. Yeah, free amino acid level goes up. And what is not shown here is also the level of micronutrients which are produced by the fungi. And the micronutrient here refers to antioxidant and vitamin, right? So this is actually the base for us to think along the line of beverage, right? Okay, so this is amino acid. You can see that the amino acid level, free amino acid level goes up. So this is one of the reasons this fermented brewer spangri can be replaced culture media because there are a lot of free amino group, right? Yeah, nitrogen group. Yeah. Okay, so this is a metabolomics analysis. You can see the changes, right, from before and after the fermentation, right? Metabolite. So as I mentioned, there are a number of micronutrients, right? So you can see vitamin, you can see antioxidant, and a lot of also useful metabolic intermediate which are produced. So this is briefly summarize the fermentation per se, right? So you can see that this traditionally brewer spent green is used for landfill and animal feed. But in Singapore, we are stuck because both have we have the constraint on both. So after fermentation, we enlarge the application range, right? Because the nutrition value is enhanced. You have amino acid, metabolic intermediate, citric acid, and then micronutrient, right? So next one is how we assess this feasibility of this brewer spent green to replace yeast culture medium. Okay, so well, this is an aim and novelty, right? Novelty is not just to replace for the sake of replacing, but also cost reduction. So we have estimated the cost reduction can be about 90%, right? Okay. Well, this is indicated here, right? All right. So the yeast extract. When we talk about yeast culture medium, there are basically two components that cost money. One is a yeast extract, the other one is a peptone, right? A peptone in particular compete with a food supply, and because it comes from plant, from meat, from casein, and so on, right? And so what we are showing here is that so the nutrient extracted from the what you see here, okay. So YPD is actually the normal yeast culture medium, and the blue one is unfermented. You can see that even with 2% glucose, the growth cannot match with the growth of using traditional culture media. The negative control is unfermented BSG because is nothing really there to support the yeast growth. The yeast that we use is the red yeast, rhodose, which produce carotenoid on itself, right? Okay. Okay. So if we use fermented BSG, you see the result actually that is actually support the growth of the yeast and producing this carotenoid intrinsically. This is a result that is more technical. You can see the control. This is a fermented. This is an unfermented. A fermented BSG media without glucose. This is a yeast culture medium in black, and then in blue we have a fermented BSG media plus 2%. So you can see that it's more or less there. But while the yeasts are growing, are they producing a similar level of carotenoid? Because this reflects the functionality of the yeast, right? So we can see that is not as high, but much better than using a no fermented BSG, right? So now we try the conventional baker's yeast Saccharomyces cerevisiae. This one is actually I did my PhD on this Saccharomyces cerevisiae, so it's very close to my heart. You can see similar result. So without glucose, right? Actually unfermented BSG with or without glucose it won't support the Saccharomyces cerevisia's growth. This is using conventional, traditional culture media, the commercial culture media. So when you replace the yeast culture media with blue, that is fermented BSG media, with 2% glucose, you can see that it actually matched the performance of yeast growth using commercial culture media. This is a metabolic profiling, metabolomics profiling, to show that there are a lot of changes using this compared to the YP media, fermented BSG also leads to a lot of changes in the metabolite profile, which I will not drill in because time is running short. So this is basically, in very brief, in very short time, I introduce you this conclusion of this application of the fermented brewer spent green to replace culture media. I think if we look at the right-hand side, it's easier than reading the left-hand side, right? Everybody can read, but the diagram may be easier to comprehend. So barley, as I mentioned, barley green will lead to germinate, and then the malted barley would then be support the yeast growth, right? You can see here, right? So these are involved in would then turn into beer manufacturing. So in this case, we use brewer spent green fermentation, and then a new media to support the yeast growth, so to replace the conventional media for yeast growth. So you can see that we actually create alternative yeast culture media. Okay, the summary is summarized here. These two I have already explained. So we have actually widened the application way for this fermented brewer spent green. And this one I have just explained. Some numbers, right? So just now we mentioned that the 100 liter of beer, making of 100 liter of beer would generate 20 kg of BSG. Okay. So so we can generate 60 liter of grow media from this 20 kg of BSG, right? And then we can generate 2.1 kilogram of cerevisia from this 60 liter of grow media, right? And for making a 100 liter of beer, we only need 4.5 gram, 4 to 7 gram of yeast. So we still have about 2.1 kg of beer, that, of the yeast that can be used to make yeast extract to sustain this closed loop circle. You can see that we are actually really into the value creation using this side stream valorization. A benefit of this study, basically we add value, right, to the unwanted side stream. And also we have set up this monitoring system, right, to evaluate this performance of fermentation. I think this will be more useful for the scale up, at the scale up stage, right? Limitation, there are always some limitations, right? So I will not drill onto this. This is what the future direction towards this side stream valorization. Traditionally, so what the top is what we know: barley to make beer, and then the BSG to landfill animal feed, right? And then here to the green is what we have. My student has created this fermentation to create a not nutrient. I think there's a spelling mistake here. Nutrient and nutrient media. Then in turn to it can be used to support the beer making because it supports the yeast growth, right? And we still have 40 to 50% of the biomass after fermentation. This is where our dear local startup friends are working together to create new product line that will be in the market very soon, I hope, right? And so this is a future direction. You can see that as a whole, we actually close the loop, right? By adding fermentation, we take nutrient out of the waste. So we actually stop this traditional direction. We create the value nutrient to look back to the initial stage. We also remove the solid waste, remaining waste into something more useful and biodegradable. This is a model called circular economy. When I talked to my colleagues in US, I wasn't sure whether it's popular. That was about five, 6 years ago. Now maybe it's slightly gaining traction, I hope. Yeah. So this is what we call extension to a circular economy. So we also publish, right? We also publish in the sort of top journals. Recently, we have just had one paper accepted to analyze this fermentation, accepted in the comprehensive review in food science and food safety. This is a journal from the IFT in Chicago. I've been there. It's a wonderful place. We also published a food security article in Trends in Food Science and Technology, another top journal. Another top journal, traditional technical journals are listed here. These are selected publications for you to refer to if there's an interest. These are some of the websites regarding our overall research activity. You may want to take a look. We have been sort of promoting our R&D activity to the media. Sometimes we talk about food security. These are my beautiful teams, education side and then the research side. As I mentioned, we started off with a very little research funding. So most of my researchers are funded by industry. And we have been very much into the media. From the US, Bloomberg News reported our views in how Singapore can enhance the food security. Last week, I received a surprise phone call from the Wall Street Journal. We had a 1-hour talk, so I hope to see the story soon, next month. We have also been interviewed, and CNN flew into Singapore to film my innovation and then push out in the Going Green program last December. We also have appeared in the Asahi Shinban in Japan, CNBC, BBC, and the AFP. So this is the end of my presentation. A little bit, I'm not sure whether it's technical enough, but I welcome all the questions. Thank you.
Awesome. Thank you so much, William. I really appreciate that. That was a fantastic presentation. We have a few questions here coming through the Q&A section. Those of you who have questions, remember to plop them there in the Q&A section for William, and we will ask all the questions until we either run out of questions or reach the end of our seminar time. So the first question for you, William, comes from Marilia. What are the species of yeast used for the fermentation of brewers' spent grains to produce enzymes?
Interesting question. Because of these companies working with us, not sure whether I'm allowed to review the species, but we can take offline. Yeah.
Sounds good, Marilia. I think that means you can email William separately.
Yeah.
Awesome. So Leonard now asks, did you pasteurize the brewer's spent grain before inoculating it?
Yes. Yes, we do. Yeah.
Awesome. And then we have another question here from Karyin. Apart from generating new media for yeast growth, what are other applications of the fermented brewer's spent grain?
This is a very interesting question. Well, there's a lot of applications. For example, in our recent partnership with a US startup, coincidentally, called Sophie's Kitchen, we have demonstrated that the fermented brewer spent grain and soybean residues can actually also replace a culture medium for protein-rich microalgae. In the process, we actually do away with the requirement for sunlight. So this is particularly interesting for Singapore because of the push for urban farming. And one of the criteria is that the urban farming should be resilient to climate change. So when we do away with sunlight and with a lower cost of this fermented medium, and by the way, the microalgae grow 3 times better using our fermented side stream solution. So we can imagine that this small size bioreactor can be actually decentralized to the household level. Then eventually one day in the near future, we can actually have a protein supply from our backyard or even kitchen. So this is one of the applications. The first one is replacement of biologic culture medium. Second one is urban farming. The third one is nutritious beverages, right? Probiotic beverages. We were approached by Singapore small and medium enterprise. They are making they wanted to move into the food space, but they make a lot of ice. So I told them it's very difficult because it's very crowded. To establish your own brand, you take time. Can you wait? They say no, they want to make money soon. So I say maybe how about we use our fermentation, this nutritious beverage, combined with your traditional brand name product. And sure enough, but that kicks off very fast. So they receive government funding for the project, and then the product will be on the market this year. Because of the COVID-19 pandemic, there's some delay. Otherwise, we'll probably already see the product on the market in Singapore. Yeah.
Cool, very exciting. You touched on cultivated meat in your last answer. Can you talk a little bit more specifically about how brewer spent grain maybe can be valorized for applications in cultivated meat or cell cultured meat?
That is an excellent question. We are doing that because one of the issue is the culture meat, the requirement for the bovine fetal serum, right? And that is a bottleneck to bring down the cost. So we are educating the cells to go vegetarian. So that we are training them to grow in the fermented side stream solution by supplementing some nutrient, because it can be actually quite a culture shock for the animal cells to just be vegetarian in a sense, right? And so this is a project that we are doing together with our colleagues from A-Star in Singapore and also New Zealand. So we are trying to create this not just ground beef or ground chicken. We are creating this beef stick on the petri dish in a cost-effective way. Yeah. So this is ongoing, to summarize.
Yeah, fantastic. I'm excited to see future research publications about that method. So a few other questions here. Adwin asks that in your presentation or in your experiment, the fermentation ends after 3 days. Is this because fermentation beyond 3 days no longer improves the nutritional value of BSG, or are there any other considerations?
After fermentation, is that right? What do we do? Oh, okay. Is it because and 3 days? I see. Well, as you know, good things don't last forever, right? So if you let it ferment, I think some toxic stuff may start emerging. We know that there's some metabolite, even aflatoxin may start emerging from the long stretch of fermentation. But for industry, why we say 3 days? We actually go beyond that, but it doesn't look as pretty as 3 days, right? And for industry, no one wants to have a 2-year fermentation. They will all collapse, I think. So the time is money, and in this case, I think we see value. 3 days is a sort of optimum condition for us to move on, because fermentation is a means to an end. It's not the, we're not really optimizing fermentation condition at this stage, but this would depend on the industry's interest and some curious mind in the academic space. So this is, in conclusion, 3 days is not random. It's optimum in our hand. It's an optimal number of days for fermentation. Yeah.
All right, thank you. And also for the sprinkling of life philosophy. Here's a question from Leonard again. Do you see any role for anaerobic digestion in your processes?
Okay. We have not tried anaerobic digestion. The idea here is that the aerobic digestion is much faster, and the anaerobic digestion tend to be slow, and then I'm not sure whether the microbe will perform equally well in terms of producing micronutrients. And since our aerobic fermentation stopped the spoilage entirely, because there's no more smell, so we don't see a strong reason to embark on the anaerobic digestion. But there may be a reason behind the question that I will be happy to discuss offline. Yeah.
Awesome, thank you. We have a question here from Lauren Blake. Are the amino acids in the fermented culture mostly free? Does the fermented BSG produce enzymes that can break down proteins?
Interesting question. Very interesting. The amino acid we have analyzed are mostly free, but this is a nature of fermentation. So they break down all the polymer into building blocks. In the case of protein, it would be amino acid, right? For enzymes, there are microbes that which produce enzymes, secrete enzymes to digest the substrate. In this case, yes, the fermentation actually of BSG, the breakdown of the BSG, sorry, the component in BSG was done by the enzyme secreted by the microbe. This is an excellent question. Sorry, I need to have a sip of water. No use to talk for so long. Sorry.
No worries. We do have you working today. You've been talking for almost a full hour already.
Yeah. Right.
Yeah. We're happy. If you need to take a break, totally happy.
No. I'm okay now. Yeah.
Sounds good. It looks like we have 5 questions to go. So someone here is asking, do you have any insights for fermenting BSG with algae instead?
Fermenting. I see. Is this I think it's a sequential fermentation, if I'm not wrong. So I think people use this to sort of recover nutrient from the general food waste. So this is ongoing with some collaborators outside Singapore. They are doing this sequential fermentation. One is using fungi, yeast. The other one is using microalgae. I don't know whether I answered the question.
Yeah, I think that's helpful. Just whether or not, it sounds like it's tangentially related to your research right now. And that there may be papers you can recommend for someone to read more on how to, ferment with algae. Okay, awesome. Here are a few more questions. One from Karyin. Is the fermented BSG edible, similar to tempeh, since the free amino acid is higher after fermentation and it might be nutritious or otherwise advantageous for a human diet?
So the question is after fermentation, whether BSG can be edible, is that right? Okay. So I would not recommend that because the idea here is we take nutrient out of the BSG. So if any consumer who want to try the BSG as a nutritious snack, um, maybe we should do it before the fermentation because after fermentation, I think maybe instead of washing off the ferment before we wash off the pH after fermentation, whether it can eat or not, right? Now we may need to change the visual aspect of the fermented BSG, but it doesn't look very appetizing. But I'm sure it's nutritious because there are a lot of micronutrient, free amino acid, and things like that. So I haven't tried that. I forgot to try that because I only tasted fermented solution. It was very tastes very good. Yeah. Okay.
All right, fantastic. Here's another question from an anonymous attendee. Unfermented BSG with glucose doesn't lead to growth, as you demonstrated in your experiment. So does that mean it's because it's inhibitory to the yeast, or why does it lead to growth?
This is a very good question. Maybe I wasn't clear when I go to the technical part. You see, that's a problem, right? In fact, the glucose has been depleted in the fermentation of BSG because the microbe, they also need the glucose to grow when they do the fermentation, right? They secrete enzyme to digest, and then the glucose would then be taken up by the microbe to grow. As a result, fermented solution has virtually no glucose, and therefore we need to add glucose for the yeast to grow when we replace a commercial yeast culture medium. It's not the glucose is inhibitory to the yeast. In fact, it's a reflection of a lack of glucose. So we need to add in 2% glucose. I think the chart was not clear on this point. Yeah.
Yeah, I think they were specifically asking, I guess, in the base case where you had the unfermented BSG, so the not yet fermented BSG, and then you had glucose, that 2%
right.
added. Why didn't that lead to growth?
Okay, so basically, when you compare to unfermented BSG with or without glucose, it will not grow because the nutrient like free amino acids, all these have not been released from the BSG. While if you replace commercial culture medium with fermented BSG plus 2% glucose, you will see growth, especially in the case of Saccharomyces cerevisiae. Yeah.
Got it. Thank you. That's helpful. So how do you preserve the fermented BSG and prevent it from deteriorating? Is it storage a challenge because the volume of fermented product is larger than the original BSG volume?
A very good question. I mentioned that the fermentation of a BSG stopped the spoilage, right? And you also reduce, for some reason, it tend to be drier, so easier to store. So I don't see any problem in terms of long-term storage. By the way, there's no need for long-term storage because our dear friend from Ultrapack is actually making this fermented BSG solid BSG into other useful product. So we will not just leave it there because that would not be a circular economy. Yeah.
Yeah, that's fair. You have a lot of people in the chat saying, thank you for such a great presentation. We have a number of questions to go through still.
Please.
A question here from Luca. Does the fermentation also produce reducible sugars by the degradation of hemicellulose or cellulose that would enable the metabolites to be used as precursors for Maillard reaction and flavor synthesis?
Wow, very interesting. Very interesting. This is something I have not thought of. I will be happy to discuss with the one who posed a question. Very happy. Yeah, very interesting. Yeah.
All right, sounds good. Sounds like that's another one we'll take offline. Here's a question from Mariano. After treating the BSG, do you need to remove any compounds that can be toxic for the microorganisms?
Okay. So this again, it was not on my mind because we just close a loop, right? Toxic or not, we just close them. Everything is in the house, right? So close a loop. So how to remove that can be toxic? We actually after treating the BSG, we more our interest is more on how to find ways to apply the solution of fermentation. Whether the microorganism is still alive or not by any toxic substances generated during fermentation was not exactly our part of our interest, but it is worth looking at. This question that worth looking at because eventually these toxic substances may also affect future application in consumer food. And so risk assessment of fermentation is an area that we are moving into because it's never everything is so fantastic. We should also look at some shortcomings, the risk assessment and toxicity from fermentation is one of the areas we are investigating. Yeah.
Great, thank you. Here's a question from Leona. Hi, Professor Chen. You mentioned that side streams like BSG spoil quite quickly. Have
Yep.
you looked at what the microbes are that are growing on the spoilage? They're proposing that maybe those are better candidates for biovalorization because they seem to grow really well without the need for supplementary glucose.
Huh. Interesting. Interesting. This is very similar to my way of doing research, right? So, you know, in academia, people tend to complicate matters. And after publishing 200 papers, I go the other way. I simplify matters. This is precisely the same approach. Instead of using the probiotic food-grade microbe, why not using the candidate to sort of ferment the BSG and without glucose, correct? The problem is that it sounds very good, but first we do not know. We have not analyzed microbe, the cause of spoilage. Second, I need to convince my student to bear with the smell because these will take the microbe that cause spoilage would make the environment very not so pleasant to be in. In this case, maybe anaerobic digestion would be the way to go, but it's a very worth thinking of investigating. Yeah, thank you for the question.
Yeah, that's a neat research project idea. Okay, we have a question here from Adwin. Do you have any examples of metabolites produced from the fermentation that affects the taste of the fermented BSG?
No. I mean, we have all the data in the paper. So if you look at the previous slide, the last slide, there's a paper from Curry, from the student, published in the Journal of Functional Food, and that is talk precisely about all these metabolites. But I'm not sure whether it has anything to do with the flavor, but we can discuss offline. Yeah.
Awesome, thank you. Yeah, again, the recording will be posted on our YouTube channel in a couple days or so you can go back to that last slide and check out William's recommended papers. There's a question here from Katsum. Is the methodology that you discussed here in your presentation being commercialized in Singapore today?
Yes. Both the fermentation is sort of being commercialized, and also how to produce the other product from the solid residue after fermentation is also being commercialized. So, yeah.
Okay, fantastic. And lastly, this question I think you covered in your presentation, but it's our last question, so maybe you have additional thoughts to add. How can you kind of give an overview of how you extracted the nutrition from the BSG?
Okay. So we are approaching this problem from the very cost-effective way. So we do the sort of overall extraction. Yeah, we do not separate different component after fermentation. So basically, it's just fermentation, and then wash out the fermented BSG, and then analyze the solution, and then find applicate way to apply this fermented solution. Yeah.
All right, that's actually our very last question for today.
Oh, okay.
Which is amazing. Do you have, I want to thank everyone for attending. Just kidding, we just got another question snuck in as I was about to wrap up. We have a question here from Andrea. Again, wanted to thank you for an interesting presentation. She wanted to ask if you think it's possible to repeat the fermentation process in another type of barley, like non-conventional beer, more crafty, and if so, how much you think the flavor can change after the fermentation process and the sugars?
Oh, yeah, it's entirely possible. So I did not highlight because it's not part of the technical details. In the big picture, the technology we developed in NTU Singapore are platform in nature, which means that we can, like for example, fermentation we apply is applicable to not just brewer spent green, but also soybean residue. And even recently, we also have applied fermentation to extract chitin and chitosan from the prawn shell. So fermentation is actually a platform technology, can be applied to a wide range of raw materials. So I'm sure your type of raw materials, think another type of barley, certainly can be used as a entry point for our platform technology. The only thing is that we may need to tweak the condition here and there, but that should not be an issue. Yeah.
All right, great. Thank you so much again to our audience for being so engaged and asking such great questions. Before we wrap up, William, do you have any final thoughts for this group of alternative protein scientists?
Okay, so I would say as I was a young one, right? So I was very much in the lab work, day in day out, and then sometimes we lose we may lose the big picture, lose sight of the big picture. So it's important to always bear in mind of the big picture, but still be passionate about what you do, and be creative, right? So don't always push towards one direction. Find ways to sort of solve a problem with alternate approaches, and there are always ways to approach the same problem. We can see that, especially in Singapore, young students tend to just study hard. But I like to point to the aspiring scientists that study smart, do research in a smart way. It can be also very productive. Right? So that's all. Yeah.
Thank you so much. We really appreciate you joining us today. It was such a joy to learn from you, William, and very excited to see your technology start to make an impact in the alternative protein field. Awesome. So thank you to everyone in our global community of scientists who joined us today. We will be back again next month with Dr. Julian McClements, who will give us an overview of how to make better plant-based milks at our normal 10 a.m. seminar time. So again, thank you so much for everyone for joining us today, and I hope you all have a good rest of your evening.